drugs that can be consumed by humans must first go through various stages of trials before receiving approval for distribution. It is no longer a secret that drugs must be tested in various stages in order to determine the resulting effects, both therapeutic effects, probation, on toxic effects, or what can also be called the treatmental effects. Stage 1 is the stage of discovery and development of new drug. In this stage, researchers carry out research that comes from new insights regarding a disease, which allow researchers to decide or make a product that can stop or reverse the effect of the disease. This most basic stage can be carried out by testing molecular compounds to find a beneficial effect on a disease. In this stage, a product or compound whose effects you want to know is tested at the molecular level so that the response that occurs at the molecular level can be assessed. Also, in this stage, researchers will carry out various experiments and find out how compounds that have the potential to become drugs are absorbed, distributed, metabolized, and secreted. Find out about the potential and mechanisms of action, the best dose if the drug is used, the best way to administer the drug, the side effects that occur when the drug is consumed, how the drug can interact with other drugs, and the level of effectiveness of a drug when compared with the same type of drug. Stage 2 is preclinical testing. In this stage, researchers must first find out whether the drug has the potential to cause a serious damage or not. There are two known types of preclinical testing, namely in vitro, which is testing in laboratory equipment such as in glass or plastic container, which resembles in vivo condition. The second type of testing in vivo. This is a type of testing on living creatures in conducting preclinical research studies. Research must provide detailed information about dose levels and toxicity. Their preclinical testing has been completed. Research review their findings and decide whether the drug can be tested on humans. Stage 3 is clinical testing. Clinical research carried out usually refers to research of trials carried out on humans. In general, research will first consider what they want to achieve for each of the different clinical research phases and start the investigation on the drug process. Before this process is complicated, clinical testing cannot be carried out. Stage 4 is about FDA approval. FDA approval will be obtained if the development stage carried out has evidence from initial tests along with preclinical and clinical research that the drug is safe and effective. The discovery and testing of new drugs aims to demonstrate the safety of the drug and also its effectiveness for use in the population study. Stage 5 and last is the stage for monitoring drug safety on the market. In drug safety monitoring, a true picture of the safety of the product can usually develop over several months, some even years. Thus, the FDA reviewed the reports of problems with prescription and over-the-counter drugs, after which it can decide to add and warn, dosage, or use information supplemented with other steps for more serious problems. Thank you for watching. Wassalamualaikum warahmatullahi wabarakatuh.